Hello, everyone, and welcome to our Immunology Pills mini webinar series. So first of all, I would like to thank you for joining this event today, and I hope you and everyone in your surroundings is safe in these challenging times we are going through. Today, we will have together a short journey through the mixed leukocyte reaction and other presentation assays. This is the agenda of today. In this presentation, I will talk about mixed leukocyte reaction, the so-called MLR, and its workflow. I will also cover the antigen-specific T-cell activation assay and the associated workflow. I will give an overview about the MDSC suppression assay as one example of suppression assays. Moreover, I will present you common challenges that are related to this assay's workflow. And at the end, I will sum up my talk. Since these assays are commonly performed in the field of immuno-oncology, I'd like you to have a look on this illustration where you observe the crosstalk between different immune cells and the tumor cells. You see here that there are several players. Some are known are effector cells, and such as T cells, NK cells, and macrophages that are able to kill tumor cells. On the other hand, we have immune cells such as Tregs, myeloid-derived suppressor cells, MDSCs. These are able to suppress immune responses, which will have a negative suppression effect on effector cells and therefore support the tumor growth. A major goal behind immuno-oncology therapies is to find ways to boost the effector responses and or diminish the suppression. However, given the complexities of the tumor microenvironment, developing such therapeutic approaches against cancer can be very challenging. So therefore, in the search for effective cancer therapy, thousands of immunotherapy compounds must undergo laboratory screening for each new drug before they are tested in humans. One of the strategies to test these compounds is via immuno-oncology assays. These are in vitro cell-based assays performed to predict compound mode of action, test downstream efficacy, and also predict immunogenicity. We know from the early pharmacology classes that MOA, or mode of action, is defined as the specific biochemical interaction through which a drug substance produces its pharmacological effects. And immunogenicity is defined as the ability of particular substance, for example, antigen or epitope, to trigger an immune response. And now we have wanted immunogenicity, where the immune therapeutic drug does its desired job to prevent the suppressive function of the suppressor cells and enhance the function of the effector cells, which will eventually eliminate the tumor cells. On the other hand, we have unwanted immunogenicity, in which the effector cells reject the drug and produce anti-drug antibodies, which will cause inactivation of the therapeutic drug, and in rare cases, even mediate side effects. So immuno-oncology assays are diverse. They come in different shapes and colors. And during this talk, I will cover MLR assay, antigen-specific T-cell assay, and MDSC suppression assay as one example of suppression assays. So some few words to mixed leukocyte reaction assay. In this assay, you simply mix cell populations isolated from allogenic donors. It is essential that the cells are isolated from allogenic donors because HLA mismatching is important for T-cell activation. The cell population mixture could be PBMC and PBMC, or T cells plus antigen-presenting cells, dendritic cells, or you simply mixed two T cells populations. You incubate the cell mixture in culture, then you add, your spices, for example, checkpoint inhibitors, and then at the end, the immune response that occurs could be measured in forms of proliferation, cytokine production, etc., etc. So the final result shall tell you something about your drug effects. This is an example of MLR workflow. It involves monocytes and T cell isolation. And for this purpose, products such as CD4, CD8, CD3, CD14 microbeads, and our technology Atomax are suitable. The assay also involves dendritic cell differentiation from monocytes where max cytokines, IL-4, and GM-CSF are essential factors for the differentiation. DC and T cells are cultured together, and here Tex-Max medium is ready to feed. And eventually, T cell proliferation, activation, etc., could be analyzed with MAX1X, MAX1 analysis using reaffinity antibodies. Now, this is a result of MLR assay showing flow cytometry analysis of enriched naive T cells co-cultured with monocytes or dendritic cells on the left, and on the right, T cell proliferation in different co-cultures is indicated. I included here a direct link to the PDF file that includes the MLR assay data in case you would like to have a look at it. Our second assay for today is antigen-specific activation. Here, antigen-presenting cells like dendritic cells are isolated, loaded with antigens, and basically mix with T cells from the same donor. You incubate the cells mixture in culture, then add your herbs, as I said, maybe checkpoint inhibitors, and at the end you measure immune response in forms of proliferation, cytokine production, and so on. Example of antigens that could be tested in this assay are cytomegalovirus antigens, super antigens, is Streptococcus enterotoxin B, SEB, or perhaps the tumor antigens. Here, I would like to show you the cytomegalovirus reactivation assay, which is basically the same principle of the previous slide. So you mix DC plus T cell plus cytomegalovirus peptide plus your test compound, and then T cell proliferation and cytokine production is measured. What I would like to emphasize here is that you could use different type of DCs for the purpose of this assay, either MoDCs or blood DCs, which are several subsets. And moreover, we have multiple options of peptide pools that can be used for antigen-specific T cell stimulation, including different peptides for SARS-CoV-2. For more insights into this, you can feel free to watch the first webinar of this series from my colleague Lorenz Fühler, and here is the link. So this is a workflow of antigen-specific assay using MoDC and T cells. Here we also need monocytes and T cell isolation. So again, different CDs, microbeads, including straight from whole blood CD4 microbeads, straight from Buffy-code CD14 microbeads, as well as CD8 and CD3 microbeads. And our technology, Multimax Cell 24, suits good here to run a large number of samples. Differentiations of DCs from monocytes with the help of max cytokines, IL-4 and GM-CSF. It's also very practical and helpful. The assay is culture-based, so again, takes max medium. For the cell proliferation analysis, max quant, max analyzer, and reaffinity antibodies are also ready. we have to keep in mind that speed is required in these assays since people process large number of samples from PBMC donor bank, meaning that this assay is automation friendly. Now, in this slide, you see some data from T cell activated by MDSC, loaded by CMV peptides. If you look at the graph on the right, you observe that either CMV peptidator or CMV protein nicely mediated T cell activation. On top, the steps you observe the steps of MDSC generation until antigen loading and T cell activation, which is basically explained. I explained on previous slides. In this slide, I would like to show you the antigen-specific T cell activation assay workflow with the option of blood DC T cell combination instead of more DC. So for the isolation of blood DC, these products can be used: CD141 microbead kit, CD1C dendritic cell isolation kit, CD304 microbead kit, the so-called the neuropilin one, as well as CD4 and CD8 microbead. So the rest of the workflow stays the same as I presented on previous slides. So we have data available showing T cell proliferation after activation by blood DC loaded by CMV peptides, which you observe here on the graph shown on the left. Just note that XPDC written on the graph refers to cross-presenting DC, and we are happy to share these data with you in case of interest. Now, in this slide, I would like to show you one of the immuno-oncology suppression assays. So MDSC suppression assay. This assay, performed as the following. One way is to isolate MDSCs from whole blood or from tumors. And another option, you can generate them from monocytes. And in this case, they are called more MDSCs. Then these isolated MDSCs can be cultured with T cells and your test compound, and then you do the rest of the classical measurement. Now, what kind of challenges do we face while working with human MDSCs? So, well, we have challenges in different parts of the workflow. So in this part of cell separation, challenges could be long isolation time, different starting materials. Some researchers want to isolate different cell types from one sample, and also the problem of unspecific cell activation. One solution we suggest here is our straight from protocol. So why? Because it's simple, fast, and reliable. You don't need any density gradient. You don't need to count or wash your cells. And the whole process from A to Z requires only 30 minutes. So this is an example of multiple separations from one sample using a straight from technology. So this technology allows multiple cell isolation of CD14 monocytes, CD8 positive T cells, and CD4 positive T cells from the same sample in just 1 hour time. So this is really convenient. But for large sample processing in immuno-oncology assay workflows, cell separation via automation is very, very practical. So Multimax Cell 24 separator nicely fits for this purpose. It has multiple practical features. For example, sample scalability. to processing of large sample volumes, for example, from leukocytes. You can do that in really in short time. Moreover, we have some researchers that encounter cell culture challenges, such as inconsistent cell culture results, suboptimal antigen stimulation, or difficult translation into clinic. So the solution for that is the standardized culture using max premium grade cytokines to optimally differentiate and mature MDSC. The exact unit per milligram dosing allows high reproducibility. So these data show the functionality of MDSC differentiated and matured using these cell culture solutions. These cells are able to increase expression of activation markers during maturation. They're able to migrate. In a migration test, they are also able to produce IL-12 upon stimulation with cytokines. We could also encounter flow analysis challenges, including unspecific staining, low reproducibility, or antigen-specific cells difficult to analyze. So the solution here are recombinantly engineered reaffinity antibodies. They consist of one universal isotype control, so human IgG1. They have high purity and greater reproducibility. Part of the workflow of the flow analysis solutions are Miltenyi Biotec flow analysis kits. So this rapid cytokine inspector, human, we have maxplex cytokine kits, human and mouse. Also have different options for immunophenotyping kits, such as 8-color immunophenotyping kit for human, and we also have a nice solution for MDSC detection kit. This is for standardized and simple flow analysis of MDSC subsets. So now to wrap up, immuno-oncology assays are essential tools that support the development of novel immunotherapeutics, and you can rely on our support on the entire workflow of antigen presentation assays and other immuno-oncology assays with simple, standardized, and optimized solutions. This is my disclaimer. And thank you for your attention.